All right, this is our week 4 project on cartilage regeneration in rabbits using different types of mesenchymal stem cells. So this is the introduction. So some background information. It is known that cartilaginous disorders comprise a wide range of diseases that affect normal joint movement as well as ear and nose shape. Mesenchymal stem cells, also referred to as MSCs, provide a promising regeneration alternative for treatment of degenerative cartilaginous disorders. So the biological question that the researchers were asking, the study is hoping to demonstrate effects of different laser-activated MCSs in regeneration of rabbit articular cartilage defect, and it's comparing the ability among the different MCSs derived from bone marrow, adipose tissue, as well as ear cartilage. These researchers are asking how these different MSCs will develop with respect to isolation, proliferation, and their restorative potential. Their hypothesis that was shown was that bone marrow derived MCSs would be shown to be most effective in regenerative ability out of the various types of stem cells. Okay, and then on to the assays and methods. So to start, they formed different groups. There were 12 adult rabbits allocated equally into 4 groups. Group 1 was their positive control group. The rabbits were suprachondrially injected with phosphate buffered saline. Group 2, the AD-MCSs, was a transplanted group. The rabbits received a 1 milliliter laser activated AD-MSCs via injection, which is from adipose tissue. Group 3 is the same thing, and their MCSs were from bone marrow tissue. And then group 4 was also very similar, with the only difference being their tissue was from the ears. And then the start of the isolation and collection of the MSCs. The isolation of the rabbit for MSCs for adipose. The rabbits were put under anesthesia, and then the bilateral inguinal adipose tissue was extracted. And then for the bone marrow MSCs, the BM MSCs were isolated and collected from the whole femur or tibia of the rabbits after euthanasia. And then the isolation of the ear MSCs. The rabbits were put under anesthesia and the cartilage was taken from a cleaned ear. And then the tissues for the 3 MSCs were then cultured. They were cultured and grown to provide adequate samples for the experiment. And then onto injection of the MSCs. MSCs were activated using a low-level laser. They were then injected into the stem cells. The MSCs were resuspended in PBS and activated using a low-level laser for 10 minutes. 1 milliliter purified stem cells with PBS were injected at a concentration of 2 × 10⁷ cells per milliliter at the excision site's margin. In the positive control group, 1 milliliter PBS was injected into the effect site just because it was a control group. And the MSC proliferation assay, growth of MSC of different sources was measured using trypan blue cell viability assay at 1, 3, 5, and 7 and 9 days after the initial MSC seeding. And then so onto stem cell characterization. They were first characterized using flow cytometry. The phenotypes of the stem cells were studied using flow cytometry. Gating was then used, forward and side scatter. Gating is used for identifying the cell population. Stem cells differentiation was then used. The MSCs were analyzed for their capacity to differentiate into adapogenic, osteogenic, and chondrogenic lineages. They were then characterized on a morphological evaluation of cartilage regeneration. Both ears of each animal was inspected, and that continued until the auricular defect was completely closed. They then did a microscopical evaluation. Tissue sections were cleared in xylene embedded in paraffin wax and sectioned at 3 to 5 micrometers thickness for histopathological and immunohistochemical examinations.
So the histopathological examination, it started with the prepared deparaffinized sections being stained with hematoxylin and eosin and Mason's trichrome and toluidine blue stains, in addition to Orchian stain that detects elastic fibers. And the stain slides were examined using a light microscope. Then an immunohistochemical examination for S100 was done. So all groups were prepared for S100 immunohistochemical examination, and each specimen's mean value and standard error of the mean were calculated and statistically analyzed. And then the statistical analysis was analyzed using a one-way analysis of variance, ANOVA by SPSS version 17.0 software to assess the significance of the mean between the groups, followed by the least significant difference, Fisher post hoc test.
Okay, results. Okay, so the first result we're going to take a look at is the MSC proliferation assay. As you can see in the graph, this showed that the adipose stem cells increased the greatest over the course of the assay in comparison with the bone marrow and ear stem cells. And this is, yeah, as I said, this graph just shows us that increase, and we can see the blue line represents the adipose stem cells, which increase the most. So for the flow cytometry test, the stem cell phenotypes resulted in positive expression of 4 of 7 surface markers. So this basically means like all 3 types of stem cells reacted in the same way. Then in the MSC differentiation test, the 3 types of stem cells were tested to see if they would be able to differentiate into the chondrogenic, adipogenic, and osteogenic differentiations. And as you can see in the chart on the right, they were able to. Now for the physical results, this is the actual test on the rabbits that we can see in the diagram on the right. The tissue was cut away from the rabbits, and the stem cells were treated, and we can see how they healed. So as we can see, the stem cells, which are the 3 columns, the first 3 columns, healed the rabbits with less scar tissue and looks like less surface damage than the control group. Now, on the microscopic level, we can see some differences in these results, as the bone marrow MSCs were able to produce more cartilage than the other type of stem cells, which is this whole research paper is about. Now, in the immunohistochemistry test, this is even more apparent that BMMSCs were able to had a higher S100 staining, which means that they were producing more cartilage. And finally, again, with gene expression, they were tested different genes and whether they were expressed in these stem cells. And as we can see from these charts, the highest level again was bone marrow stem cells.
So now to the interpretations the authors made. So the results were interpreted and as pointing to ADMSCs having the highest proliferation rate, with the bone marrow MSCs having the highest cartilage differentiation potential. And this means that ADMSCs had the highest efficiency in tissue regeneration, yet the bone marrow MSCs achieved similar tissue regeneration results and had the greater impact on regeneration of cartilage, making them the more effective as a stem cell type. And so based on this data and the reasoning behind their interpretation, our group agrees with the findings and interpretation of the researchers. And then the conclusion is that bone marrow mesenchymal stem cells had the highest proliferation rate and chrondogenetic potential when compared to adipose-derived stem cells and ear stem cells. It had better reactivity to the S100 protein, and it had higher production of COL2, aggregine, and TGF-B1. And so bone marrow mesenchymal stem cells could be better than adipose-derived mesenchymal stem cells and ear mesenchymal stem cells for the regeneration of cartilaginous defects. And then some future ideas which our group had was using human mesenchymal stem cells to test which have the best regenerative effects, comparing how bone marrow mesenchymal cells compare to other types of mesenchymal cells in rabbits other than just the adipose-derived and ear-derived, and then comparing how bone marrow mesenchymal cells, or testing if they can be used for regeneration, for treatment of regenerating defects other than cartilaginous. And then just our citation was the research paper which we used.
All right. Thank you for your time. This was the end of our presentation.
Thank you.